I'm Dr. M. Parimela Celia, Assistant Professor, Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous Cuddalore, Tamil Nadu. I'm going to present a topic Helicobacter pylori, a bacterium which is responsible for causing the disease peptic ulcer. Let's see the introduction. The human stomach is considered as an inhospitable environment for growth of most of the bacteria. But there is the spiral shaped organism which has been occasionally visualized in the gastric mucosal layer. But there is no evidence for the association of disease symptoms. This organism is classified first as Campylobacter pylori and now it is called as Helicobacter pylori. And other species of this Helicobacter have been so far isolated from stomach and intestine of other animals. Marshall and Warren cultured this organism from human gastric mucosa and they show the association of this organism with gastric inflammation, that is the swelling. And it is a spiral shaped bacterium that colonize the gastric mucosa. And this etiological agent, that is the causative agent, is associated with the disease gastritis, that is inflammation of gastron, that is stomach, and cause peptic ulcer. This organism colonizes nearly 50% of the individuals. In this picture, you can see the structure of this Helicobacter pylori. It is a spiral shaped organism which shows the polar flagella. Regarding the morphology, it is a gram-negative bacteria which shows helical shape, that is spiral or curve, and it shows 0.5 to 1 micrometer in width as well as 2.5 to 5 micrometer in length, and it is blunted or having the rounded ends while observing in the gastric biopsy specimen, and the cells become rod-like and coccoid on prolonged culture. That is, it shows pleomorphism and it shows the unipolar flagella. Regarding the cultural and biochemical characters, this organism can grow well at optimum temperature 37 degrees centigrade, and it grows well in the media such as chocolate agar as well as Campylobacter media, and it grows under the microaerophilic conditions, and the growth can be enhanced if we add 5 to 20% carbon dioxide. Regarding the biochemical features, this organism shows positive result for oxidase, negative result for catalase, and strongly positive result for urease. Now we will see the pathogenesis. This organism, Helicobacter pylori, colonizes the gastric mucosal layer, and this will spread mainly through the oral contact, that is through the mouth, and it spread through oral fecal route. And the main reason for the spreading of this infection is overcrowding as well as the poverty. That is, the people, if they are crowded in an environment, there is the possibility for spreading of the infection will be more, and the infection is said to be mild to acute, that is severe gastritis, and it is associated with gastric antrum, that is gastric metaplasia. Gastric metaplasia, it is just the cancerous thing that is found in stomach cancer. Any part of the stomach can be involved. It colonizes the mucosal layer, but they do not penetrate the mucosa. It colonizes the mucosal lining of the stomach as well as the duodenum in case of man as well as in animals. Adherent to the gastric surface epithelium or pitch epithelial cells deep within the mucosal crypts adjacent to the gastric mucosal cells, they may found. Then the mucosa protect the stomach wall from its own gastric digestive enzymes and hydrochloric acid. The mucosa also protects this helicobacter from the immune response. Most gastric adenocarcinomas and lymphomas are concurrent with or preceded by an infection with Helicobacter pylori. That is, this organism, if it is identified in the gastron, there is the possibility for the development of gastric cancer as well as the cancerous growth in the lymphatic system. And now we will see the virulent factors which is associated with this Helicobacter pylori. So the presence of these factors will give virulence, that is the pathogenicity to this bacteria. First one, the presence of polar sheathed flagella. This flagella will exhibit a type of motility called corkscrew motility, which enables the penetration into the viscous environment, that is the mucus, because the stomach, it is having the mucus lining. In addition, it possesses the virulent factors like adhesin, hemagglutinin, sialic acid binding adhesin, as well as Lewis blood group adhesin. It produces the enzyme mucinase. The role of this mucinase is to degrade the gastric mucus and cause localized tissue damage. And it produces the enzyme urease, which converts this urea into carbon dioxide as well as ammonia. This will neutralize the local acid environment as well as they cause tissue damage. Also, it produces acid inhibitory protein and cause tissue damage. In addition, it produces the vacuolating cytotoxin. Cytotoxin damage the cells, especially the epithelial cells. In addition, it produces invasins, hemolysin, phospholipase, alcohol dehydrogenase, and they will protect this from phagocytosis as well as the intracellular killing of pathogen. Now we will see the indications for non-invasive testing for Helicobacter pylori. If a person is having the following symptoms, it is strongly recommended to test that particular person for Helicobacter pylori. And now we will see one by one. First one, dyspepsia. Dyspepsia is the symptom that is abdominal discomfort. If a person is having abdominal discomfort, either bloating or burning sensation, they are strongly recommended to test for Helicobacter pylori, as well as the history of active peptic ulcer disease. That is, if they have any family members having this ulcerous peptic ulcer, the chances for getting infection is more. So they may be advised to test for Helicobacter pylori. Then the next one is gastric malt lymphoma, that is mucosa associated lymphoid tissue. So they will be having such sort of symptoms. They are recommended to test for Helicobacter pylori, as well as following gastric cancer resection. If the person is suspected to have gastric cancer, they are advisable to test for this test. Then following peptic ulcer surgery, if a person undergone surgery, they also recommended. So first degree relative with gastric cancer, if a person is having gastric cancer, and they are recommended to have such test. And also long-term non-steroidal anti-inflammatory drug therapy. And now we will see the laboratory diagnosis. And now this infection can be diagnosed by both invasive and non-invasive test. In case of invasive, we have to go for the endoscopic test, that is the biopsy. By doing endoscopy, that is the device called endoscope, which will be inserted through the oral cavity, and it will be inserted into the gastron, and the biopsy, a tissue section has to be taken, and it will be observed under the microscope, and the pathogen may be isolated and it will be stored in cultures, and it is further strained by specific strains, as well as by doing gram staining we can identify the morphology, and also we can do biopsy testing for urease detection in urea medium. And now we shall see the diagnosis by non-invasive methods. First one, serological analysis. In this, the blood sample has to be collected from the patient and the serum has to be collected, and we have to do ELISA test, enzyme linked immunosorbent assay, and we have to do this urea breath test in case of patients suspected to have such infection. They are asked to swallow the urea solution. In this test, the patient drink urea solution labeled with an isotope carbon. If Helicobacter pylori is present in the urea, it will be converted to ammonia and carbon dioxide, and that can be measured in the breath. And now let's see the treatment. The treatment can be made by using specific antibiotics, bismuth salt, and ingestion of bismuth salicylate. And antibiotics such as tetracycline, metronidazole is recommended for 2 weeks. And also use of omeprazole is recommended, as well as clarithromycin. Thank you.